Hello everyone, I'm Mr. Furlong, and today we're talking protein synthesis. In this process of protein synthesis, which is the process of making proteins, we're going to be bringing in a lot of information that we've been talking about this past year and putting it together to take a look at how proteins are made, an extremely important process that occurs in all living things. Now we know what DNA does. It stores our hereditary information, it gives us our traits, it controls everything that's going on inside the cell, but it needs some help, and it's getting that from RNA. Remember, RNA is ribonucleic acid. There were some basic differences between RNA and DNA. RNA single-stranded, DNA double-stranded. RNA has ribose, DNA has deoxyribose. RNA has the base uracil, which is abbreviated with the letter U. DNA has thymine, which is abbreviated with the letter T. There are three different kinds of RNA, and they each have a different job to do. The first type is called messenger RNA, or we abbreviate with a lowercase m and a capital RNA. The job of messenger RNA is to carry the information from the DNA out to the ribosomes where the proteins are made. Recall that DNA can't leave the nucleus, and so this is carrying the message, if you will, of DNA to the ribosomes. The second type is called ribosomal RNA, or a lowercase r, capital RNA. And in ribosomal RNA, this is going to be involved in helping to make up the ribosomes. This is, of course, where proteins are going to be made. And the third type is transfer RNA, which is abbreviated with a lowercase t and a capital RNA. This is going to transfer the amino acids from the foods that we've eaten to the ribosome so that we can assemble them in the proper sequence. And we'll see that this is going to be very important in order for our protein to be able to work. One thing to take note of on the transfer RNA molecule, there's a series of 3 nucleotides right here at the bottom. We'll go 1, 2, and 3. These are called the anticodons. But DNA and RNA working together is what's going to make this protein. So let's use an analogy of protein synthesis. Let's say that we're building something. Before we build it, we want to have some plans. We're going to make up some blueprints, or at least someone's going to be making up some blueprints. That has all the specifications needed to make that object. In this case, we're going to be making some type of a vehicle. Blueprints are analogous to the DNA. However, when something is being built, we're not going to take the original blueprints out to the place where it's going to be built. So we'll have to make a copy of that. So out comes a copy of these blueprints. That's sort of like the messenger RNA, if you will. So this goes out to the job site. Well, now people have to put this vehicle together. And so a bunch of people are coming to assemble the product. These are sort of like the transfer RNA. They're bringing the know-how, but they're also bringing the supplies needed to put that vehicle together. And when they're done, ooh, that's a nice looking vehicle. Ooh. In this process of protein synthesis, there are 2 major things going on. The first is called RNA transcription. Now, as you can see in the word transcription, the root word is scribe, which means to write. So we're going to be writing an RNA molecule from the DNA. And although we're going to talk about messenger RNA right now, all 3 types of RNA are made through this process of transcription. So if you take a look at this diagram, we have the RNA polymerase enzyme is going to unzip just a small part of the DNA molecule. And it's going to put those nucleotides in place to form a messenger RNA. Now on the messenger RNA, there's a series of 3 nucleotides that we're going to refer to as a codon. Remember, there was an anticodon on the transfer RNA. On the messenger RNA, we have codons. Let's take a look at how this process occurs. As I'm going through this program, which is from the Concord Consortium, you're going to see the steps appearing on your screen. Make sure you write those down in your notes. So here we see a bunch of cells. So there's our cell. The big purple thing in the middle is the nucleus. And we want to make a protein. So we have to go through this process of transcription. So let's get a closer look. There's our chromosomes. There's the DNA. Let's get a close-up look at the DNA. And remember, during transcription, we're going to need to have an enzyme, the RNA polymerase enzyme, come along and unzip the DNA. Only one side of the DNA molecule is going to be used. That is referred to as the template strand. Now remember that DNA has thymine in it. And of course, thymine always pairs with adenine. But our next nucleotide is adenine. Adenine, remember, pairs with thymine, as I just said. But there's no thymine in RNA. Remember, uracil takes its place. So a uracil will combine with the adenine. Cytosine, as before, pairs with guanine, and guanine with cytosine. And this will continue one at a time until that RNA molecule has been made. A's with U's, C's with G's. At some point, it's going to reach the end of that gene, and the messenger RNA has been made. Once the messenger RNA molecule has been made, it will detach from the DNA. DNA zips back up, and the messenger RNA heads on out to the ribosomes, where translation will take place. Translation is our second stage in the process. Here we're going to arrange the amino acids in order to make that protein. Now it's called translation because what we're really doing is we're translating that code of the DNA that was given to RNA and messenger RNA, and we're going to translate that in order to put the specific amino acids in the right place in order for this protein to do its job. Ooh. Well, let's take a look at this process of translation, again, using the Concordium Consortium website to do this. Recall that on the messenger RNA, every 3 bases forms a codon. In this case, the first codon is AUG. Now, this codes for an amino acid, but which one? Well, scientists years ago was able to break this genetic code. We don't need to worry about how they did it, but we do need to understand how can we interpret this code. So you have a chart like this. So you see that we have a column that says first base, second base, and third base. So we're not talking baseball here. We're talking about the bases on the RNA nucleotides. We look to see, well, what is that first base in the codon? In the first one, it was A. The second one, it was U. And so we look to see, well, where is A and U? It's going to be one of these 4. So then we look at the third base, which is down here, G. And it tells us that, oh, this is the methionine amino acid. And if you look real close, it says start right behind it. This is what's known as the start codon. If you look closely, there's no other start codon. That means that every single protein begins with this codon, the start codon. So how do we get methionine there? Well, this is where transfer RNA comes along. Remember, transfer RNA has the anticodon. The anticodon for the codon AUG is UAC. So it knows it's in the right place. And look what it just brought us, the methionine amino acid. The next codon is CCA. So if you look on your chart, CCA codes for proline. And so along comes another transfer RNA. Notice its anticodon GGU is matching up with the CCA. And look what automatically happened then. Methionine formed a peptide bond with the proline. The next codon is GGC. Again, if we look on our paper, that's glycine. Peptide bond forms, but that first transfer RNA takes off then. That can be used over and over again. So it's going to go out into the cytoplasm and pick up another methionine amino acid. The messenger RNA continues to move along the ribosome. The next codon is GGC, again, which is another glycine, and the peptide bond forms, previous transfer RNA leaves. And this is just going to happen over and over and over again. GAG codes for glutamine. AGC codes for serine. Again, previous transfer RNA leaves, and as the peptide bond forms. Notice what's happening. We're forming a chain of amino acids. Now remember, some amino acids, they don't want to be in contact with water. Other amino acids, they want to be in contact with other amino acids. And so they're constantly shifting around and so on. The UUG codes for leucine, and our protein continues to get bigger. CUA, again, is another code for leucine. AUU is ileoleucine. Notice how all these amino acids, we just use the 3-letter abbreviation for them. The GGC is glycine. AAU is leucine again. And so again, this process is happening over and over again, and we're slowly building our protein. But something just happened here. The last codon, UAG. All right, so if we look on our paper, what does it say? It says stop. That's what's known as the stop codon. So this is how the transfer RNAs know to stop adding more amino acids. The protein has been made, and it's taken on its 3-dimensional shape. It's actually a very fascinating process, and without this process, and life as we know it would not exist. So I hope you enjoyed watching this video. I know I had fun making it. I don't like that sign off. Let's do something else. Ooh.